Hello everyone, welcome to Life Science with Veena. Today we are going to start with one interesting topic, that is instrumentation. And in this instrumentation unit, we are going to first talk about a technique called as electrophoresis. Okay, we are going to talk about electrophoresis. So, what is electrophoresis? If you split this word into three portions, that is electro, phore and cis. So electro is electric field, phore means pores, and cis is separation. So it is a separation technique. Now separation of what? Separation of biomolecules like DNA, RNA, proteins under the influence of electric field through the pores on the basis of their mass, charge and size is called as electrophoresis. So here is a simple definition of electrophoresis, that is, it is a separation of biomolecules like DNA, RNA and proteins under the influence of electric field through the pores on the basis of their mass, charge, size, etc. Okay. Now, as I said, the charge is the factor that affects the migration of the molecule. So, the movement of the molecule is based on, or the charged molecule is based on the charge of the molecule. For example, DNA is negatively charged due to the presence of phosphate group into it, right? So the negatively charged molecules migrate towards the positively charged electrode, that is anode, and if the positively charged molecule is there, then it will move towards the cathode, which is a negatively charged electrode. Okay, so basically it is the migration towards the opposite electrode. Okay, and the rate of migration will depend on the charge, first thing. Second, it will be dependent on the size, molecular weight, shape, strength of the electric field, the type of the gel, whether it is agarose gel or a polyacrylamide gel. Now, if you talk about the migration rate, the smaller the molecule, it will move faster. For example, in a crowded train, the person who is very thin can board very quickly, whereas a person who is obese may find some hindrance while boarding into the crowded train. Similarly, the smaller the molecule, faster it is going to move, and the molecule which is larger in size, having a large molecular mass, will travel slowly. Okay, so smaller will move faster and farther, whereas the molecule which is large size, it will move slower and will cover a less distance, okay, as compared to the small size molecule. Okay, now here you can see that here the mixture of DNA molecules of different sizes. Here are the wells. It is loaded into the wells, and this is the direction of migration. So the longer molecules are at the top of the gel, whereas the shorter molecules are at the bottom of the gel. Okay, so this is how the migration of a biomolecule DNA will take place over here. Now if you talk about the types of electrophoresis, they are gel electrophoresis, zone electrophoresis, free flow electrophoresis, capillary electrophoresis, etc. But for a time being, for this presentation, we will be only restricting ourselves to the gel electrophoresis, and particularly in this presentation, we'll be talking about agarose gel electrophoresis and SDS polyacrylamide gel electrophoresis. So let us see one by one. Now, Here are some nice images from my lab where I have conducted the practical electrophoresis, agarose gel electrophoresis, separation of DNA molecules, and for separation of proteins, that is page I have conducted. So this is H, and this is a vertical apparatus of page, polyacrylamide gel electrophoresis. This is also a page. This is also a page. This is also a page. Only the first one is of Agarose gel electrophoresis, which is a horizontal gel electrophoresis. Here you can see there is a electrophoretic apparatus, and it is connected with what? It is connected with the power pack, that is the electric supply. Now here you can see in the second image there are wells, and these wells, in these wells the sample is loaded, and after applying the electric current the molecules will start migrating. Okay. This is a, in the fourth image you are able to see that it is a dual gel apparatus, means at a time you can run two gels. Okay, so this is how it works, vertical and horizontal. Now if you talk about the equipments, so these are some equipments like gel casting, gel casting tray is there, then power supply, for power supply power pack is there, this is a lead, these are the electrodes, these are the combs for making the wells. Okay, now this is again an image of agarose gel electrophoresis, and this is how after separation the DNA molecules will look like. The bands will appear orange because of the staining solution, that is ethidium bromide, which will intercalate between the DNA bases, and that is why the bands are orange in color. And we have used over here a UV transilluminator for UV light for visualization of the DNA bands. Now why it is blue in color? Because of the loading dye, we can say, or a tracking dye, that is bromophenol blue. Okay, that is why the color of the sample is blue, because it is a tracking or loading dye, bromophenol blue, and the staining dye is ethidium bromide. So this is how the bands of the DNA look like. This is how the loading will occur. With the help of a micropipette, we can load the sample into the wells which are formed with the help of comb. Okay. Now let us move on to the components of agarose gel electrophoresis. So there are power cables, as I said, cathode and anode electrodes, tank lead is there, casting dams are there, gel tray is there, electrodes are there, buffer tank is there. So these are the components of agarose gel electrophoresis apparatus. Now, two reservoirs of buffer and all these things we have already seen, so no need to talk about this. Let us move on to why we prefer to use agarose only for the horizontal gel electrophoresis, because it is a linear carbohydrate polymer which is extracted from seaweed or agar bios, and it is a polysaccharide basically, and it forms a porous matrix like a sieve. Okay, like a sieve, like this. Okay, sieve, so it will form a sieve of different pore size. The molecules of different pore size can molecules of different size can pass through these pores, which are variable in diameter. So the shift it will shift from random coil in a solution to structure in which the chains are bundled into the double helices, and this is how the mesh is formed due to the formation of double helices. So this is how the agarose is going to form a network, okay, a mesh-like structure or a molecular sieve, and through this molecular sieve the DNA molecules are going to migrate once we apply the electric field. Okay. Now the second component is buffer. So whenever we watch a movie, we come to know that one is a main lead role. The lead role is agarose, and here the second is buffer, is the another component, second component. Now why buffer is used? Because buffer will provide the ions for conducting the electric current. Okay, and it will maintain the pH during the electrophoresis. Okay, so the two types of buffers are commonly used: tris acetate buffer and tris borate EDTA buffer. Okay, we use both to prepare the gel and running buffer into the electrophoretic chamber. Okay, so for it is mixed with the buffers are mixed with the agarose, and for while casting the gel, the buffer is used, as well as into the tray also the buffer is poured. It as because the gel should be submerged into the buffer in order to carry the electric current. Okay. Third is gel casting tray and the comb. So gel casting tray and comb, here the comb is shown and the gel casting tray. So casting tray will hold the molten agarose because it is going to polymerize. It is going to polymerize into a gel. So the molten agarose will be poured into the tray, and it will as we pour the idli mixture into the idli mold. Similarly, it is a casting tray and there are combs. So combs are going to create the wells into the gel where the DNA samples are loaded. Okay. Now, the fourth component is DNA sample. First, we will isolate the DNA by using some standard protocol, and then it will be mixed with the loading dye, that is bromophenol blue, and it will be loaded. The DNA sample will be loaded. Now, what is the loading dye? The function of the loading dye, it will add density to the sample, as DNA is not that dense, so sample can sink into the well, it can fit into the well properly, and generally it is bromophenol blue or xylene cyanol. It will provide the tracking dye to monitor the run. Means where our DNA molecules has reached, or is our gel running or not. For tracking that, for monitoring that, we have to add a tracking dye or a loading dye called as bromophenol blue generally, and xylene cyanol or xylene cyanol. Sixth is DNA ladder or marker. If you want to find out a specific weight of your DNA fragment, then you can, in well number 1, you can add the molecular marker whose molecular weight is known to you, and you can estimate the size of your unknown sample of the DNA bands. So what is marker? A mixture of known sizes, or DNA fragments, a mixture of DNA fragments of known sizes. So taking that as a reference, you can determine the weight of the DNA bands. Now, the fourth one is electrophoretic chamber or tank. Here you can see the blue colored tank is there, which will hold the running buffer and the gel, and it has a electrode which will set up an electric field. Now, the last one is a power supply. So power supply, it applies a voltage across the gel, so electric current basically, and so that the negatively charged DNA molecules can migrate towards the opposite electrode, that is a positive electrode. Here you can see black to black and red to red is connected, cathode to cathode, anode to anode. Now here you can see that DNA. Now it comes to, after running the gel, we have to stain it, or generally in vertical electrophoresis, the staining is done after the running of the gel. But in case of agarose gel electrophoresis, usually whenever we are preparing the DNA samples, we add a DNA stain into it, and also in the tank buffer also we add a staining solution into it, that is ethidium bromide. So after running, we don't stain the gel in horizontal, we stain it before itself, we put the staining solution before itself, and when the molecules of DNA will migrate, then the staining solution, that is ethidium bromide, will intercalate between the nucleotide bases like AT and GC. So intercalate between the ethidium bromide will intercalate between AT and GC when the gel is running. So staining solution is not, the gel is not stained after the running, but rather it is mixed with the buffer or with the samples. So the commonly used staining solutions are ethidium bromide, cyanogen bromide, acridine orange sometimes. It is added to the gel or used in the stain, used to stain the gel after running, sometimes, very rarely, but I have always done it. I have always mixed the staining solution with the sample, or with the what, or with the buffer. Okay. Now, after running the gel, we need to visualize the gel. Okay, for visualization, because the DNA bands will not be visible with the naked eye, so we need to observe the stained DNA bands. Okay, we need to observe the stained DNA bands with the help of a UV light, that is, and the instrument used for that is UV trans eliminator. Now this particular apparatus has the UV tubes inside. Okay, and the blue light imager or a gel documentation system also you can use, but generally UV trans eliminator is used for the visualization of the DNA bands. And as I shown you earlier also, so these are the UV tubes inside the apparatus, UV trans eliminator, and the bands are orange in color. Okay, so this is how the gel will look like and after the visualization. Okay, so first procedure is set up the gel, load the buffer, load your sample, DNA sample, then run the gel and stain and look with the visualize with the UV light. This is the common thing which is seen. Now the application is generally for DNA fragment separation or analysis of PCR products or restriction fragment analysis, etc. are there applications. DNA fingerprinting also has application. Checking the plasmid DNA to analyze it, whether it is nicked or linear, we can use it. RNA analysis also we can use it, and purification of DNA fragments also we can check with it. So these are the applications. In the next part we'll be talking about polyacrylamide gel electrophoresis. And thanks for watching. Stay tuned for watching page. Okay, thank you.